Hello everyone, so today we're going to talk about an important topic in biology, which is DNA packaging. Okay, so how does the DNA packages inside the cell? So as you know, it is like, so the DNA is about 2 meters long. So how can this 2 meters long DNA be fit into the small nucleus of the cell? So how can that go into the small nucleus of the cell? Well, there's 4 steps to DNA packaging. Okay, so let's start with the first one. So the first one is very simple. So that's the DNA itself. So that's the DNA double helix. Okay, so you have your DNA double helix. That's by itself. So you have here. Let's draw that's double helix, right? So this is your double helix itself. So that's the long chain of double helix. So that was the level 1. So that's level 1 when it's not. So it's in this state right here. So this is the same as that. Okay, so it is not coiled yet. So that's called level 1. So let me say here, levels, and that's going to be level 1. So level 2 is a very important one, which is the nucleosomes. Okay, so nucleosomes. So essentially, what happens is that the DNA is gonna wrap around the proteins called histones. Okay, so you have your DNA, and there's these special things called histones. Now histones, 5, 6, 7, 8. So these are histones, right? And then so histones are essentially proteins that are made up of 8 the core. So there is so here's first, let's say, talk about the DNA itself. Well, the DNA wraps around the histone twice. Okay, wraps around the histone, and that's so there's multiple of them. All right, so let's say like here. So these are multiple of these connected together in all of these cases, right? So there's are multiple of them connected. Oops, cut this one out. So let's say you have these here connected with this one, and let's just add one more just for the fun of it. So just add one more here. Okay, I'll delete that. So this, let's say that's your histone. So that's your histones in red, and the DNA wraps around it very tightly, and those forms what is called a nucleosome. So nucleosome is when you have a histone, a DNA wrapped around the histone. Okay, so this is what is formed. Now it's very important to know that the diameter is 10 nanometers. Okay, the diameter is 10 nanometers in there, and that is your nucleosome stage. So it wraps around, very important, and you need to understand, like, when further is that the histone is made up of 8 subunits. So as you know, for proteins, when it's folding up together, so histone is made of 8 subunits. Some proteins are made of 4, but it is a protein that is made up of 8 subunits. Just write this down here, maybe 8 subunits. The next level of DNA packaging is chromatin fiber. Chromatin fiber. Okay, so that is your chromatin fiber. Now what happens here is you have the bunch of the histones wrapped together. So let's say that you have your histones here, histone, 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 all of them, right? Because I've enlarged them. So here I was zoomed in. Now I'm going to enlarge a little bit. And then I'm going to have my DNA wrapping all around these. Now this is the structure that we have. We have a chromatin fiber. Now if we know from here to here, the diameter is 30 to 40 nanometers. Okay? And from here to here, as we know from before, it's going to be about 10 nanometers. Okay, so this is the chromatin. So that's the chromatin fiber. it is shortened by about, so here, so when we talk about here, when we talk about the DNA itself, here it was shortened by about 7 folds. Okay, here when we talk about the next stage, it is shortened for about 42 folds. So it is shortened by 42 folds. All right, the fourth stage that we have here, the fourth stage is called chromatin, looped domains. Okay, so chromatin looped domains. So how does that work? Well, we have the lining right here. So it forms this shape. Okay, so these here are just a magnification of what was here. So we've magnified, and that's what we got, right? So we've got from there, and now this is shortened by about 750 folds. This is shortened by 750 folds. From when we talk about the height of here, from here to here, that's about 300 nanometers, right? And it's from there. Now we have the last stage. So as we said, so this is stage 1, 2, 3, 4. We have stage 5. I'm just going to write it down here. So level 5 of DNA is called the heterochromatin. Okay, you have your heterochromatin. Now what happens is still, now it's flipping all around itself. And we have this shape right here. Now this shape is that. All right, so if I want to just say, so this shape here is this one right here. So it is this, so we've magnified again. All right, so from there, so here's that shape, or let's just put it this way. So there's that shape, right? And then keeps coiling and coiling around. This here, from here to here, is about 700 nanometers, so it's getting bigger and bigger, right? So this 100 nanometers, and then from here, it comes to the shape that we all know it. So about like, say, chromosome is something like this. Is something like this, let's say, right? So we know from here to here, now the drawing doesn't show it very well, but this is 1400 nanometers. Okay, 1400 nanometers, and that is your heterochromatin. Now one last thing to point out before we leave. So the first 4 stages, so we're talking from 1 to 4, so levels 1 to 4, okay, are called euchromatin, in which the DNA is still active, right? So it can be accessed by proteins and stuff, so it's still active. However, level 5, okay, it is condensed. Okay, so DNA is condensed beyond the looped. So let's say condensed. Okay, so it's condensed beyond the loop domains. Okay, so it is inactive. Okay, so it is considered rendered. So it's not as active as it was. Okay, so here's the notes, and hope this was helpful. Thanks so much, and have a good day.